Hello everyone, and a very good afternoon to each one of you present with us today. This is L. Shalini, representing Ryan Publisher, and I'm glad to welcome you all to the third and last day of the 3-day webinar series on nanotechnology, How Small Can We Go? Futuristic Insight of Nanotechnology. The topic for today's session is Advanced On-Body Sensor Devices for Active Healthcare. I would like to thank you all for your active participation. Kindly post your questions or comments in the chat box to make this session more interactive. Our speaker would be glad to answer your questions at the end of the session. My special thanks to Dr. P.F. Jeffy, the program organizer, Mr. Bipin Joseph, the chairman of Ryan Publishers, and Dr. Rainey Christina, the program convener. Now, without further ado, I would like to introduce our valuable resource person, Dr. Ganesh Kumar Mani. Welcome, sir. Dr. Ganesh Kumar Mani, a research scientist at Nanotechnology Center, Tokai University, Japan. He has completed his PhD in Nano Sensors Lab at Center for Nanotechnology and Advanced Biomaterials and School of Electrical and Electronic Engineering, Shastra University, Tanjore, India, in 2015. He also has won the Best PhD Dissertation Award for his doctoral thesis work. He has also participated and organized national and international conferences and presented posters in several countries. As a part of his research, he visited various countries like Korea, Germany, Thailand, and Sweden to present his research results. He is also one of the inventors in 2 patents titled Low Concentration Ammonia Vapor Sensor, Acetaldehyde Sensor using zinc oxide nanoplatelet. Recently, he received the Best Annual Presentation Award from the Japan Society of Precision Engineering for his work on microneedle sensors. He has published over 45 research papers in reputed international journals with a cumulative impact factor of over 100, with a few papers under review. He is also a co-investigator of a research project in Japan for the development of a mouthpiece pH sensor with a research budget of nearly 3 million Japanese yen. His current research interests are fabrication and development of nanostructured, nanospheres, nanorods, nanowires, nanoplatelets, nanosheets, thin film-based gas or chemical sensors for predicting food quality, developing thin film-based pH sensors for biomedical applications, and developing painless microneedles for multifunction applications as well. I would now like to request Dr. Ganesh Kumar M. to please take over this session. Over to sir.
Thank you, ma'am. Thank you for your introduction. It's really great to be speaking with you, all of you today. Myself, Ganesh Kumar Mani. I'm from Micron Technology Center, Tokai University, Japan. Today's our session topic is about advanced on-body sensor devices for active healthcare. So, first of all, my special thanks goes to the Ryan Publishers and the other supporters. I know webinar is a bit boring, so I made this presentation with a more pictorial representation than technical ones, so that everyone can have some messages take away. So throughout the presentation, the chat box will be managed by the organizing committee. So if you have any doubts or questions, maybe you can put your questions there. At the end of the session, I can answer you all. Right now, I have everyone so put in mute. Even I can turn off camera, that may distract you from listening. Okay, I hope my screen is visible for everyone.
Yes, sir.
born and brought up in Tamil Nadu, India. I finished my PhD in Sastra University, Tanjavur in 2015. In the same year, I joined as a postdoctoral fellow in Micron Nanotechnology Center with an excellent team of researchers. I have started my research with thin film gas sensors, and I expand my research area into microneedles, flexible electronic devices, and point-of-care devices. These are the awards and honors I have received in the past few years, and these are the countries I have visited for conferences and research collaborations. So here, let me introduce Tokai University. Our university was established in 1942 by Shigeshi Matsume. It has several campuses throughout Japan. The total number of students is 30,000 and the employment rate is 98%. I have to say two interesting things about our university. First one is, till today we won 37 Olympic medals. And another thing is making the solar car. Our team Tokai has won many world titles, including World Solar Challenge in Australia and Sasol Solar Challenge in South Africa. Apart from this, we have several research centers in various campuses for marine, climate change, space, and agriculture. And this is Micron Nanotechnology Center where I am working for. This is an overview about Micron Nanotechnology Center. The center comprises of 7 faculty members, and each one of them is specialized in different research field that makes them stronger to explore more. Say for example, our team contains experts from physics, chemistry, biomedical engineering, precision engineering, mechanical engineering, and even genetic engineering too. So here is the office space for common discussion and other meetings. The center was established with the concept of making freestanding nanosheet for biomedical applications. Later in this presentation, I will explain about that. For the mass manufacturing of the freestanding nanosheet, we use roll-to-roll machine, and we set up the advanced imaging center jointly with Nikon to develop new imaging tools. Now let's enter into today's technical session. The key topic of today's session is an overview about healthcare technologies and why it's so important to create point-of-care devices, and how our economy can be improved through the point-of-care devices. And we'll speak about painless microneedling, which was inspired by the mosquito bite. As I said before, our research center key material is nanosheet, so we'll explain about the electronic skin. Finally, the importance of microfluidic devices can be seen. Around the world, every healthcare system is struggling with rising cost and uneven quality. Healthcare leaders and policymakers have tried countless incremental fixes, but one thing is sure, healthcare systems today may be better than ever. If you are affected by a disease, you have to be completely rare in the following major categories. Each system can operate from different environments and complex too. Always, these 5 major things is connected closely to each other. For example, there is no use of visiting doctor's office without any laboratory test. Even the laboratory test finds some disease in your body, again you have to trust the pharmacy to get the right medicine, because you cannot take care of yourself without any medical facilities at your home. Because of the issues in healthcare system, a new point-of-care device-based diagnostics arise. Currently, routine diagnostic tests often rely on quantitative analysis of various biomarkers in a central laboratory. With that, you can get results only after several hours or sometimes even days. To minimize the time, point-of-care devices originally designed to allow medical testing as near patient or beside with less discomfort and quick analysis. The increasing demand of consumers to monitor their own health is increasing rapidly. Use of wearable technology has more than tripled in the last 4 years. This growing demand for wearable has generated a booming market. The future of wearable devices shows no sign of slowing down. You can see here how the medical equipment has evolved in the last 200 years. Everything comes portable, compact, and highly accurate. The key objective of point-of-care testing is to generate a result quickly, so that appropriate treatment can be given at right time to help life-saving decisions. In remote places, access to such kind of healthcare facilities is typically limited. In such environment, these kind of point-of-care devices are beneficial in terms of their use. Any diagnostic device must be accurate and precise and provide robust results. So the recent inventions proved that the new point-of-care devices are accurate and precise. Apart from personal needs, overall, how India can be benefited by point-of-care devices. India ranked number 2 in medical tourism, and India's health capital is Chennai. Minimum 150 patients are coming from abroad every day. So why they are coming here? The reason is some treatments may not legal in their country, and high cost and long waiting time. Due to this one, they are coming here to assess the latest technology, and we have the best surgeons in the world, low cost and low waiting time. For example, here is the cost comparison of selected surgeries. For heart bypass, so minimum they have to pay $1,30,000. But in India, they can done it in a very cheap cost of $10,000. So what is the key material for developing various point of care devices? So that is biomarker. Or in some other word, we can say as a target analytes. So what is biomarker? Say for example, if you are having a sweat and you are the sweat contains many ions, and if you are measuring the ion concentration, say for sodium, chlorine and potassium, so each ion can act as a biomarker for particular diseases. That's what we have different kinds of biomarker, like a predisposition biomarker, screening, staging, prediction, prognostic and reoccurrence. So each and every stage there will be some biomarker which can reveal your disease in your body. In the case of predisposition biomarker, they can analyze your data through the DNA level so that they can predict your hereditary disease. And in the case of screening biomarker, they can screen, that is the initial stage of any diagnosis, and staging biomarker is at what stage your disease has now. And prediction biomarker and what stage it will go after the staging, and prognostic, what is the treatment, and reoccurrence, whether after curing the disease whether it will reoccur or not. There are different kinds of biomarkers. So say for small molecules, proteins, cell count and fluids. So everything has a biomarker we can call. So we can analyze and we can predict the disease. See here inside the cell there are a lot of components. If you clearly analyze the components, and each one can act as a biomarker, and from that each component you can easily predict the disease. To construct point of care devices, we have to rely on these three key materials, that is silicon and its derivatives, metal and metal oxides, and polymers. So silicon has high mechanical reliability, high performance and IC compatibility. And metal oxides are often used as the key material to sense the biomarkers, and polymers are often used as a substrates. So it has the high biocompatibility even to your skin, low cost and single use devices, and the surface can be easily modified for anything, for any application. So coming to the first topic, painless microneedle. I should highlight the word painless here. I will tell you the reason why. So before tell you the reason, I would like to give you a brief introduction about biomimicking. So what is biomimicking? Scientists are always inspired by nature. Similarly, they try to mimic their properties. I hope everyone knows about the lotus leaf effect. The leaf surface always repel the water due to its hydrophobicity. So that's why the idea is taken from the lotus leaf, and scientists are artificially created anti-weight coatings, say for the t-shirt and anti-weight smartphones and even the shoe sprays and windshield. Not only the lotus leaf, there are several examples now. Say for example, kingfisher bat, boxfish and shark skin. So Japanese bullet train is inspired from the kingfisher bat. Due to this aerodynamics, the scientists analyzed the flying structure and its aerodynamics clearly, and then they designed similarly to reduce the drag so that the train can go faster. Even the same scientists done for the boxfish. And using the shark skin, they analyzed the shark skin completely and its aerodynamics, and then finally the same like structure can be used in the flight wings and even the swimmer's cap. So now come to the point, why microneedle? Nearly 20% of a global population is affected by needle phobia. That means those people are afraid to take the injection because of the pain. So a usual procedure in medical industry is to take any fluid from your body is through the injection. There is no other option. That's why if they are not allowing to put the injection, so the doctors cannot analyze their disease level. And even the later stage, it is quite difficult to diagnose the disease. That's why annually 1.3 million deaths occurs. So what we are going to do here, what we are going to mimic. So here we are trying to mimic the mosquito bite. So you know the mosquito, right? It often sucks blood from your body and giving with a very less pain. That's why we are trying to mimic how the mosquito gets blood from your body. That's why we are doing some analysis here. And here you can see only the female mosquito bites. So it extracts 1.9 microliter only for 2 minutes. So to mimic such kind of system, first we need to understand their mechanism. For that, we have fixed camera at different angles, and we are analyzing how the mosquito sucks blood from your body. So how fast it can, and what are the things it is doing in your body. So first see here, you can see the mosquito sitting on the skin here and trying to suck blood from the skin. So here is on the other angle, you can see how the mosquito bite. So you can see the blood veins very clearly, and it is puncturing it. On here, you can see how in the lateral direction you can see how it is making the hole in your skin and getting the blood. So you can see here in this one that it is not making a hole directly. So if you closely watch this one, you can find it is giving some vibrations first, because the mosquito's labium is very soft, so it cannot puncture your skin directly. So it needs to give some vibration to make your skin rigid, and then it can puncture. You can see in this video very clearly. As a next step, what we have done is we have taken the SEM of the mosquito and its labium. So first thing, we just tried its mechanical property, how it is making a hole. But here we want to exactly mimic the same size, so that which is having the pain almost same as the mosquito bite. That's why here you can see it is a mosquito's labium, which is having the length around 3.5 to 4 millimeter, and the diameter is 30 micrometer. It often changes 25 to 30 micrometer. So you can see the commercial needle here, it is having 900 micrometer, but the mosquito's labium is only 30 micrometer. So here we can conclude that if the size is less and the pain is also less, that's why many people are trying to make a microneedle for the painless application, so that people can easily take the drug through in a painless way. Here you can see the human skin cross section. So here the both microneedle as well as the commercial needle is punctured into your skin, and you can see the length, how long it travels into your skin. In the case of microneedle, it is only 10 to 15 micrometer below your skin. But in the commercial needle, it goes few millimeter into your skin, so that it can go and touch your nerve endings. In the case of microneedle, it cannot touch the nerve ending, so you cannot feel the pain. But in commercial needle, it go and disturb the pain nerves, and you can feel the pain more. So there are different kinds of microneedle available in the market. There is a solid microneedle, coated microneedle, dissolving microneedle, and even the hollow microneedle. What we are trying to mimic here is the hollow microneedle, which is same as the mosquito bite. So here you can see the mosquito's labium is 25 to 30 micrometer, and here this is a titanium microneedle what we made in our laboratory using sputtering technique. The sputtering, what we done is we can take the copper wire with the same diameter of the mosquito's labium, say 25 micrometer, and then we coated with the titanium through sputtering. We have some customized equipment to perform this action. So after the sputtering, what we do is we etch and remove the copper wire, so that you can get this hollow structure which is almost similar like the mosquito's labium. So here you can see the comparison of the commercial needle as well as the titanium microneedle. So we fabricated the microneedle. As in the first step itself, I was telling that it is a painless microneedle. How to evaluate the pain? So till today, there is no analytical technique which can evaluate the pain. So indirectly you can evaluate. That is usually we can say as an alpha amylase test. That is an enzyme. If you feel stress or pain, the amylase activity in your saliva gets more. So initially we start with the experiment with mice. What we done is, so you can see the microneedle, we are puncturing it onto the mice leg, and immediately after puncture we take the saliva, and then we do the alpha amylase test and how much alpha amylase present in the mice saliva after injection and before injection, and then we analyze it. So what we found is using this microneedle, same as the mosquito's mimic microneedle, has lesser activity of alpha amylase. That indicates that the mice is not stressed much, so it did not feel much pain compared to the normal needle. Then we can try, we tried with different sizes of microneedle, say for 50 micrometer, 100 micrometer, 200 micrometer. And what we finally found is, if your microneedle is less than 95 micrometer, which is almost same as the mosquito's labium effect. But if the limit exceeds 95 micrometer, which is almost same as the commercial usual needle. So that's why we limited our research work to 95 micrometer only. As a first step, we confirmed the pain evaluation
elevation. As a second step, we wanted to confirm its hollowness. So we inserted a small micrometer level glass tube into this artificial titanium microneedle, and we passed the water. And you can see the water coming is on the other end. So it confirmed that the complete structure is hollow and without broke, because you can see there is no leak here. So using this microneedle, we developed a wristwatch type device for the glucose sensor. So we put the glucose sensor inside this device, and for the glucose sensor, your blood can be extracted through this titanium microneedle. So there are 4 titanium microneedle fixed here, and we can, using this negative pressure, you can easily suck the blood from your skin, and then it can analyze in this wristwatch how much insulin you needed, and then it can supply the insulin to your body. Here I have to highlight how important it is to develop microneedle based sensor. So here you can see current status of microneedle sensor as per the Scopus database. So till today, using the microneedle as a keyword, you can get 12,000 patent result and only 3,200 document results. But in the case, if you see the sensor, in the results it's more than 1 million. But when you combine these two, microneedle and sensor, you can get only 250 document and 3,815 patients. So it can say that there is a lot of research have to be done to create various kind of microneedle sensor. That's why we dedicated our research to develop a single cell pH sensor. So why pH is important here? So the average human cell is in the size of 5 to 100 micrometer. The cells, each cell can maintain the intracellular pH in the range of 7.1 to 7.2. But if you analyze the cancer cell, it is maintaining the intracellular pH with 6.2 to 6.9. So with this small difference, if you test the pH of intracellular fluid inside the cell, you can identify between the normal cell and cancer cell. That's what we wanted to do. That's why we developed a single cell pH sensor which can easily classify between normal and cancer cells. So this research may not be used directly on your body, but this kind of research is highly wanted by the pharmaceutical people. Say for example, if some company wanted to develop some drug, there is only one technique. They can culture the cell, they can give the drug, and then they can see the result. So in this case, millions and millions of cells can share their drug, and then they can evaluate. So they are collecting an average data. They don't know how each cell is responding to particular drugs. That's why this kind of pH sensor can help to develop new drugs in an effective way in a shorter time. So already there are some single cell pH sensors are available. So the problem is that one is, so they are using some nano pipetting technology or some fiber optic cell microprobe for intracellular single cell pH measurement, and even the photoacoustic thermometry. So these techniques are relying on quantum dots, pH sensitive dyes, and chitosan functionalization and cytotoxicity, and fast photo bleaching. Due to this reason, you cannot directly assess the cell to identify their pH. You always have to modify the cell with something, and then it affects their result. That's why we wanted to do the direct real-time measurement. So here we fabricated a single cell sensor as a 2 electrode. One is the AGAG cell and another one micro electrode is coated with the zinc oxide. And then we put into the fish shape. We always begin the test with fish shape after the laboratory experiment. First, after putting into the fish shape, we artificially modified their environment by adding a small amount of citric acid and large amount of citric acid. And then the pH value is calculated separately, and we inserted this microneedle into the fish shape and measured the pH again. And then it has the good linearity, and sensitivity range is 45 millivolt per pH. But the ideal pH sensitivity is 58 millivolt per pH. But even though in this micro level, the sensitivity is a good achievement for us, so we are going for the next level for the animal testing. So here, we wanted to try the mice cerebrospinal fluid pH. So usually during operation, especially during the brain operation, the brain cell pH often changing. So they have to monitor. So at this moment, there is no technique they can measure it directly. So they always measure every 5 minutes or every 2 minutes, they can measure the pH. So if these kind of micro electrode have developed, they can use it along with some other devices. They can instantaneously record the pH during the operation. So if they see some abnormalities, definitely they can stop or they can take some actions. So here what we have done is the mice is given anesthesia and then we inserted the microneedle onto the mice brain and we checked the pH level. So the first graph here it shows the laboratory experiment. So we purchased some pH buffer solution and tested our needle electrode and we get the result. That is for the reference. And then we run the same experiment on the mice brain. So by comparison, we can get the pH data. Here is the comparison you can see. The yellow one is measured by the commercial pH meter and the blue one is measured by the pH electrode was developed by us. And you can see the pH range. The yellow one, it shows the pH 7.13 and the blue one, that is a Micron electrode, it shows the pH of 7.03. So there is only 85% only the data is correct, because when we insert the microneedle into the mice brain, the sensor sensitivity is slightly affected by the brain tissues and other fluids. So we are trying to recover the problem so that in future we can get some 100% accuracy to work this pH electrode as successful. Not only this one, there are many people working as the microneedle array patch. So our specialty is creating a single microneedle, but many researchers are developing such kind of microneedle patches so that they can deliver the drug easily. For delivering the drug, the microneedle array will be useful. But for extracting something from your body, single microneedle is the better way. Coming to the next part, electronic skin. So that's usually made up of nanosheets. So here, already people are very afraid to taking medication because of the injection. And even for monitoring something, people are very afraid. So here, what is electronic skin? It looks like same as tattoo. So you can stick on your skin and it's almost similar like tattoo. It will not hurt you anymore and it will not create any inflammation or irritating the skin. But it can measure some activity from your body. Say for example, electrical muscle activity, body temperature, pH, and even the skin vapors it can measure. You can see how the bearable market can grow each year by year. So it is gradually increasing. So people have started using these kind of devices to monitor their biological signals. So almost the electronic skin looks like this. We developed such kind of skin to measure the skin pH to detect from heat stroke. So how to measure the heat stroke? So say for example, blood, saliva and tears have minimally invasive or highly invasive test. But sweat, if you do some exercise, you can get your sweat. And even if you are going to affect by the heat stroke, your body produces more sweat so that we can easily identify the dehydration level from your skin sweat. That's what we have an idea to measure the skin sweat using this artificial nanosheet. That is we used to call as a electronic skin. So why the electronic skin is important to measure using sweat-based system? So when you use the electronic skin-based system, it can require minimal electrical components. So the electrical waste can be much reduced. And it is a very low cost and you can dispose it easily, even biodegradable. So here you can see the model of the electronic skin-based pH sensor. So this is the production method. We use the sacrificing layer method. So initially we take the silicon oxide and we deposit with calcium cellulose acetate as a sacrificing layer and then PDMS as a substrate. And then finally, after depositing the electrodes here, you can see the sensing electrodes here is the reference electrode is the AgIO3, which is our own lab developed material, and SB2O3 is a working electrode. So after depositing, we can sacrifice the layer to make the nanosheet as a freestanding, and then you can transfer on your skin. And when you connect it with the electric components, it can measure your pH. So here, the sensor is transferred to the artificial forearm, and even after several bending and twisting, the nanosheet have withstand its ability. It did not tear and visibly no crack have been found. And then we tested with the pH measurements here using a standard procedure in the laboratory. So first we tried to measure with the standard pH buffer solution and we estimated the sensitivity about 74. And then even after 3 trials, the sensitivity is same and the sensor having good stability too. After that, when I say about the heat stroke, when the person have the tattoo and going outside for a walk in a sunlight. So the sun temperature can affect the sensitivity of the sensor. So we try to do the temperature dependent testing with artificially. We have the thermometer, digital infrared thermometer, and here you can see the nano sheet is kept on my hand, and then the temperature is increasing using the dryer. And even after several temperature settings, room temperature and 34.7 and 34.8, the sensor did not affected by the external temperature. So it is just measuring the skin sweat pH directly without any external stimuli. And the paper was recently published in ACS sensor, and even it appeared as a cover image too. And this is the pamphlet we have developed to distribute through in our university, titled as Your Health in Your Hands, Novel Wearable Technology Can Easily Detect Heat Stroke. Because in Japan, every summer, there is a lot of people are affected by the heat stroke. Even last year summer, more than 40,000 people have taken to hospital due to this heat stroke. That's why we got this research grant, and we are developing such kind of electronic skin-based system to detect heat stroke. Not only heat stroke, even we can detect the dehydration level of the athletes too. As I said before, our Tokai University alone have won 37 Olympic medals. You can have we use this device. It was running in the saliva, it can measure the pH, and we can measure dehydration. If the dehydration is there, it can alert the person and ask them to take some matter to take. So here, is very good study. Here is lab student asked him to be a black coffee. So we have several waters based on the black coffee is 5.6 and is 2 points, and then after that we take difference in 3 different waters. So here I showed only one, but we put the places like near the teeth, near the canals, and near the body. Which part of the how this and the near the dark has the best study. And here we see the changes because the green one is the back, the red one is the front teeth, and the blue is the canines. So the higher the 3 different traits, you can easily differentiate the null one after reaching the previous one. So this one we can use some based devices, and then we can use it as to smartphone, and then it can say that it takes to many lives. Coming to the next topic, microneedling. So the appropriate device, very low quantity of sample for multiple analysis. For example, in the microfluidic device, with 1 micron, you can say how much platelets you have and whether your blood can contain any circulating tumor cells or what is the platelet count. So it can do the multiple functions at the same time. On the other hand, it did not need any reagent consumptions, very low reagent consumptions for the analysis, and the analysis time is very, very short, and it can be disposable and easily makeable. So the reduced overall cost will be reduced for each analysis. And even the next level, they are trying to develop organ on a chip. So say for example, your kidney, so it is doing some purification function. So in the same case, if they create a same model, so it can be used as instead of the organ transplant, we can use this microfluidic chip to do the same function as kidney do. So how it can be used in the recent days. So metastasis, right? The cancer cells. So here you can see the primary cell, and through your bloodstream, the primary cell can transfer to the another place, and it can create the secondary tumor. The cancer is not a single disease. It can multiply the cells into various parts of the body, and then the person will be easily die and it cannot be cured. That's why if you find the metastasis process, that is the circulating tumor cells in your bloodstream at the early stage, so it is very easy to cure. That's why we are trying to use the microfluidic device to identify the circulating tumor cells. This is the simple microfluidic device what we have created in the laboratory to identify the circulating tumor cells. As I said before, the normal cell and cancer cell have different pH level. So with this idea, we are trying to identify the cancer cell and normal cell in the bloodstream through this microfluidic device. So here, we have deposited the pH sensing material on the base, and then we covered with the microfluidics channel, which is having the size around 140 micrometer. And then when you pass through something through here, the pH of the particular location can be measured. So this is the device fabrication process. In this one, the microfluidic channel can be created using the commercial photolithographic process, but even you can use some other techniques too. So here, this is the experimental model. So here is the syringe pump, and here is the microfluidic device, and this is the outlet. So initially in the syringe pump, we put the standard pH buffer solution and taken the reference value. You can see the results here. In dry condition, it is not showing any potential difference. But once the liquid passes through and makes a connection between the two electrodes, the potential is increasing and getting stable after 30 seconds. So we can say the stabilization time is 30 seconds. After that, there is no change. So we can say, using this kind of sensor, so after 30 seconds, if you measure the potential difference, you can easily identify the pH. So as a next step, what we have done is, we taken different kinds of cells. Say for example, human lung cancer cell, rat muscle cell, and dog kidney cell. Although all three cells have different pH. And then we passed through these three different kind of cells through the same microfluidic device, and we checked the potential difference. So here you can see the black and red. The black is just the medium. Always you cannot keep the cells in the outer surface. So always you have to keep the cells in some other medium to make it live. That's why the DMEM is the medium which can use to store the cells. And then DMEM plus A7R5, that is the rat muscle cell, and then dog kidney cell and human lung cancer cell. When you pass these three different kind of cells to the microfluidic channel, so you can take the potential difference, and with the red color, that is A7R5 and DMEM did not show any change. But the lung cancer cell and dog kidney cell, the potential is shifted. And when you measure the pH using this potential difference, you can easily identify the potential difference between the cells. And then approximately you can measure the pH also. So you can find when you pass different kind of cell through this device, actually you can measure the pH also. So using pH, you can identify the circulating tumor cells also in the future. That's our prime idea. Even there are many ways to create the microfluidic device in your home itself. There is a xerography based microfluidic device fabrication. Maybe easily you can Google it. That is with a simple cutter, and then using some simple paper based disposable microfluidic device also you can create in your home with just filter paper and the crayons. Maybe usually you can purchase it easily, and then you can draw the structure and make it a heater, and the crayon is a wax, so it can make hydrophobic coating on the filter paper. Rest of the areas will be the hydrophilic, so you can easily pass something, so other areas cannot be affected. So finally, first thing, microneedle sensor, and with this setup, you can easily identify the pH sensor. On the second topic, I have say as the nano sheet, that is the electronic skin. Even using the electronic skin, you can measure the skin dehydration as well as the athlete's dehydration. And the third one, microfluidic, for the cancer cell detection. So for all these three techniques, we just developed a single pH sensor and used in three different platforms for different application. And then as a next step, we wanted to use the microneedling technology to detect the neuron membrane potential. So that is the one of the recent hot research for the artificial human brain interface. Further, we are going for the next level to develop some sensor to measure the neuron imbalance. And finally, we are also trying to overcome the current challenges, because what we have done the skin-based study is only for the adult. But the recent report says that adult skin and the children's skin both are having some dissimilarities. So both are not same. That's why we have to do all our experiment with the children's skin structure to confirm the experiment to make it commercialized. Last but not least, I would like to give some fellowship opportunities at Japan. So the first one is Maximum International Foundation offers research fellowship for the who are completed PhD in the recent years, and they are willing to take or do research in Japanese university for 3 to 6 months with a good fellowship. I'm the JSPS postdoctoral fellowship. I'm also the JSPS fellow who was selected 2 years before. And another one is the JSPS Rotanku program. It is only for the doctoral students. Those who are doing doctoral studies in India, and they can come to Japan and jointly work in the Japanese university, and you can earn the dual degree. You can get the degree in India as also in Japan. Also, you can get the good amount of fellowship, and the Japanese government will not ask you to pay for the tuition fee here. And another one is the Mixed Scholarship Research Grant. So this is directly you can do the PhD in Japan under the Mixed Grant. So now the call for Mixed Grant is open. Maybe you can try in internet and find out.
Thank you. Sorry. Yeah, maybe that's it from my side. Maybe if you have any questions, maybe you can ask now.
The session is now open for interaction. Participants can post their comments or questions in the chat box.
So any questions from the audience?
Sir, yes, sir. Can I mute and speak to you, sir?
Yes, please.
Sir, I am Shanmuga Priya, sir.
Okay.
Sir, I am completed BE Biomedical Engineering, sir. Now 2020 batch, sir.
Okay.
I am learning N4 Japanese, sir.
Okay.
Now, I don't know how to get a job in Japan. I have a internship. I completed a 1-year internship, sir, in CIT Center of Excellence. MHR is sponsored.
Okay. So you wanted to do your masters in Japan?
Sir, no, sir. For job only, sir.
Only for job. I think there are many websites available to search job. If you completed N4, maybe N5 or N4, maybe it's easy to get a job here.
Oh, okay, sir.
Maybe I will refer you some links.
Yes, sir. So please
Okay.
refer. Sir, can I mail you, sir?
Yes, please. I'll give you my
Yes, sir.
mail address in the chat box.
Yes, sir.
Okay, maybe you can give an email. Okay, thank you.
Thank you, sir.
Participants can feel free to share your comments or to ask questions if any. Dr. Ganesh has shared his email ID in the chat box. Kindly refer to it. I would now like to invite Mr. Bipin Joseph, Chairman of the Ryan Publishers, to convey the vote of Thanks. I'm sorry. Sorry to interrupt, sir. I guess there's a question again from Mangala Gauri.
Okay. How does this nanosheet indicate the results? So actually, there are two ways. Maybe we can take some connections and use the measurement unit separately. And the other thing, we can use the color. We used to say as a colorimetric nanosheet. So if the temperature rises, the nanosheet color will change. So usually it will be transparent if the body temperature increases, and then it will become opaque, so that you can nicely identify. So without any external devices, only with a nanosheet, you can easily decide to take the results from that. Okay, any other questions?
If there are any other questions, we are still waiting for that.
Hello, sir.
Yes.
Sir, regarding this nanosheets, I have a doubt. Actually, only we can monitor the pH level, or even we can detect the blood pressure or the heart rate through these nanosheets, sir. Because you said it's used for athletes and other people who are under dehydration or whatever it is. If you are using it for an athlete, definitely there will be some changes in the heart rate, and which may be fatal to some people. Because of dehydration or whatever it is. So these nanosheets can be used for measuring these blood pressure or any changes due to this cardiac problems or anything like that, sir?
Yes, we can. So our first success is to make the pH sensor. Now we are measuring the blood pressure using the same kind of nanosheet. We are trying to manage the blood pressure also. So as of now, we did not succeed because the nanosheet, as I said, the thickness, it is only 60 nanometer, 60 to 100 nanometer. Handling such type of a very thin nanosheet is quite difficult. That's why initially we thought of using applying sweat is very easy, because easily you can transfer the nanosheet onto your hand and then you can sweat easily. But in the case of pressure, heartbeat or something, we need to accurately measure the pressure. The blood pressure. So that's why it takes some more long time. Maybe in 1 or 2 years we will be succeeded. Already we are starting doing it.
Okay, sir. Okay, sir. Thank you so much, sir. Your presentation is very useful, sir. Thank you.
Thank you.
We are still waiting for more questions.
I think no questions from the audience.
I would now like to invite Vipin sir again to join us.
Thank you, Ms. Shalini. First of all, I would like to thank Dr. Reni Christina for arranging this the entire event and providing us with 3 dynamic resource persons. Thank you, Dr. Reni. And then our first resource person, I thank him very much, Dr. Vinod Kumar V, who gave a talk on innovation for a better tomorrow. Dr. Madhurantakam Sasya, who gave a talk on introduction to the world of nano and its application. And today's resource person, Dr. Ganesh Kumar Mani, for giving a talk on advanced on-body sensor. I would also like to thank Dr. P.F. Steffi, the organizing secretary, for arranging this event. And I would like to give a special thank to Ms. Shalini for doing a lot of hard work. She has been throughout this session, sitting there, reading the comments and being the master of ceremony. Thank you, Ms. Shalini. And I would my special gratitude to the participants who turned up at such an odd hour, because you can understand three resource persons belong to India, but they are right now are in different countries, different time zones, so they have to manage their time limitations. They have their limitations work, and thank you all the participants to turn up in this afternoon, and let's hope we'll meet you again in our future events. And special thanks to those participants who have been with us right from the beginning, because some of them have been with us almost in every webinar. So I thank them personally. On behalf of Rhine Publishers, I thank all the resource person, the program convener, the organizing secretary, participants, and the technical advisor to make this event a success. Thank you very much. Over to you, Michelle.
Thank you, Vipin sir, for addressing our participants. And I would just like to inform you all that the feedback link for the webinar series has been posted in the chat box. Kindly do make note of it to receive your e-certificates. And thank you so much all for participating and being a part of this webinar series. And a special thanks to our resource person and to our chairman and to the organizing people involved for making this webinar a success. Thank you everyone. Have a great day ahead.